Greetings to everyone. First of all, I would like to thank the organizers of this conference for inviting us to present our topic, Half a Century of Eucheuma Kappaphycus Farming, Half a Century of Maladies. What's the missing link? My name is Danilo B. Largo. I am from the Department of Biology of the University of San Carlos in Cebu City, Philippines. With me is Flower Misoya from the Department of Botany, University of Dar es Salaam, based in Zanzibar, United Republic of Tanzania. Farming of Eucheuma Kappaphycus started in the Philippines way back in 1960s. Since then, it has spread to almost all corners of the world. From materials brought from the Philippines to Japan, it was also introduced into Brazil by some scientists who test planted Kappaphycus in the warm waters of the Brazilian latitudes near the equator. From there, cultivars went to as far as the Latin American countries of Venezuela, Nicaragua, and nearby countries in this region. Materials also went to Africa by some companies um, with buying stations in these countries, first to Madagascar and then to Tanzania. Some materials which were brought to the neighboring island or neighboring countries of Malaysia, Indonesia, as well as to Vietnam and then to India. Some cultivars were also brought to the South Pacific islands of Fiji and Kiribati and probably other Pacific island countries. In this presentation, we will look into the present state of affairs of the Eucheuma farming industry from circa 1960 to the present period, focusing on the maladies that continue to hound the farmers and the Karaginan processing industry, especially in this era of global warming and climate change. The farming of Eucheuma started in the Philippines when the original Karaginan source, the iris moss or Chondrus crispus, became overexploited. The late Professor Maxwell Doty of University of Hawaii was commissioned by FMC Marine Colloids Corporation and came to the Philippines together with the Bureau of Fisheries and Aquatic Resources of the Philippine government, plant tested um, Eucheuma in the southern Philippines of Mindanao. And there they were able to grow Eucheuma using the fixed bottom monoline method, uh, which is then the prototype of farming in the Philippines. Since then, it has spread to other parts of the country in Calatagan, Batangas, in Luzon, as well as in the Visayas, in the Nahon Reef in Bohol Island. As early as 1970s, the incidence of ice disease was already reported by Professor Gavino Trono of the University of the Philippines, then also by Professor Oyenko of the same university in 1981, who suspected the role of microorganisms in the development of ice disease. The ice disease indeed is not just microbial in nature, but has also ecophysiological precedents of stressful conditions to the seaweed sallows. My own studies during my PhD dissertation work elucidating ice disease um, made use of the ecophysiological stressors of environmental factors like high temperature, high light intensity, low salinity, which predisposes the seaweed to opportunistic pathogens like bacteria. In this study, we use both Kappaphycus alvarezi and Eucheuma denticulatum. Similarly, Solis et al. In 2010 found marine-derived fungi as opportunistic pathogens when seaweed was also subjected to stressor of low salinity. Some other scientists from Indonesia discovered bacteria as causative agent of the ice disease, especially during warm months in the Indonesian seaweed farms. India as well observed ice disease and attributed the condition to some microbial organisms, especially bacteria. So it's important that we understand the mechanism by which the organism can inflict the ice disease. In my own experiment, I simulated the different environmental factors using in temperature, light intensity, and salinity using this aquatron. Uh, by by increasing the temperature, water temperature, the cultivars of Kappaphycus alvarezi uh, turned to ice condition after several days of exposure, as shown here in the top photo, letter A. Then, when seaweed cultivars were exposed to low light intensity, it also led to the condition of ice, as shown in the middle photo here. Then, when exposed to low salinity, ice condition was also produced in the same cultivars after several days in the aquatron. Now, the right graph here shows the daily growth rate of Kappaphycus alvarezi and Eucheuma denticulatum of two strains, green and brown strain, subjected to different levels of salinity from the normal of 35 PSU down to 15 PSU. As the salinity decreased, so are the daily growth rates of these two species of eucheumatoids. That at salinity of 20 PSU, the lowest growth rate was observed. At 15 PSU, there was a false increase in growth rate, mainly because of tissue damage, wherein water was absorbed by the seaweed, causing or resulting to uh, an increase in weight of the cultivars. SI disease occurred when when the cultivars were subjected at 20 to 15 after 4 to 5 weeks of incubation in the aquatron. So it's clear here that uh, extreme environmental factors alone can already um, cause as condition uh, on leukematoids. So let's see how opportunistic pathogens can also contribute to ice development. And we have identified bacteria. In my own study, I identified a strain of Vibrio. I call it Vibrio spp11, which is a gram-negative bacterium, catalase positive, oxidase positive, and agar lysing, and motile with unipolar flagellum. The researchers in Indonesia, Sia Fitri et al., also found three species of bacteria, namely Alteromonas macleoidae, Pseudoalteromonas isakinkunai, Aurantomonas coralicida, which are gram-negative, can digest gelatin and starch, can utilize cellulobiose, D-fructose, D-glucose, D-mannose, A-lactose, and mannose. On the other hand, fungi, marine-derived fungi, was also found by Solis et al. In 2010 to be a causative agent of ice disease. These are Aspergillus ocratius, Aspergillus tyreus, and Puma sp, under class Ascomycota. Now, the media in Indonesia and Philippines took note of the observations of the scientists that seaweed farms have been afflicted with ice condition and has seriously damaged the industry in these two countries. And the scientists attributed this to the warming seas, that high sea surface temperatures cause widespread occurrence of ice disease. And so the production of the carrageenophytes went down in both countries in recent years. Um, the traditional monoclonal vegetative propagation method uh, is under threat of rising water temperature due to global warming. As you can see in this picture, cultivars on a fixed line cannot adjust to the levels of seawater during low tide, at which time sea surface temperature could be at extremely high level, and it could lead to the conditions of ice, as shown here in the lower photos. So it means that um, the fixed off-bottom monoline method is no longer a viable method. It's compromising the seaweed cultivars in most farms in the Philippines. I propose a model of the seaweed microbe pathosystem wherein seaweed is normally a substrate for microorganisms to colonize. And in the case of Kappaphycus alvarezi, um, they tend to uh, um, deploy an arsenal of antimicrobial agents, which could ward off the early colonization of microorganisms such as opportunistic pathogens. But when seaweed cultivars are under extreme environmental conditions, this could stress the seaweed that could uh, make it susceptible to infection by the opportunistic pathogens, leading to the occurrence of ice disease. This observation of antimicrobial agent was found in my own study in the 1990s, uh, which was an unpublished work during my dissertation work in Japan. So it's clear that microbes can play an important role in ice development together with uh, environmental factors that could lead to um, the condition known as ice. Now, in my study, I was maintaining Kappaphycus alvarezi in a bay in Japan near the university where I was studying. So I had monthly harvesting of the cultivars from late summer up to early autumn or late autumn at the onset of winter, so that seaweed thalli are on various conditions, health conditions. During summer, they are healthy, but during late autumn, they become ice disease. And during the other months, uh, they could have unhealthy conditions. And we try to extract antimicrobial compounds from these dried materials, and we found out that indeed there was antimicrobial agent from this seaweed. When we use this to challenge different bacterial strains that were originally isolated from same seaweed, we found that young thalli of Kappaphycus alvarezi, young and old thalli, which remained healthy, can actually inhibit um, almost half of the bacterial strains. There were about 20 bacterial strains that we isolated. So it inhibited about half of them uh, using the extracts from Kappaphycus alvarezi. Extracts from other months of cultivars collected from other months, especially during unhealthy conditions, uh, although it inhibited the bacterial strains, but at a lower degree. So less than 20% of these strains were inhibited. Compared to standard antibiotics, our extracts from the seaweeds is comparable to that of penicillin as well as polymyxin B, but it's lower compared to streptomycin and kanamycin. So it's very clear here that there is an antimicrobial activity in seaweeds, especially when they are still healthy. So further on to the different strains of bacteria that were challenged by the seaweed extract, we found various reactions of the different bacterial strains to the extract, depending on when they were collected. So in months where they remained healthy, even during late summer in September, most of the bacterial strains were inhibited by the extract. Whereas during winter, from as early as October to December, our extract uh, were not able to inhibit the growth of most bacterial strains. So it explains that at healthy conditions, antimicrobial agent is produced, but during unhealthy conditions, our cultivars were no longer producing much of the antimicrobial agents. Now, this picture shows an agar plate of Vibrio SPP11 um, with the diffu- uh, with discs that were immersed in the seaweed extract. So in this duplicate samples, we observed a zone of inhibition around discs with the extract, but none could be observed in discs that were immersed only on the solvent. So here, as the control. So in the other strains of bacteria, um, some other strains could be inhibited by the commercial antibiotics, especially this strain of bacterium called P57 and P100, which were inhibited mostly by the antibiotics, as well as these fish pathogens, Pseudomonas fluorescens, originally from fish that was donated to us by another laboratory in our university. So it clearly shows that antimicrobial agent can be produced by our eucheumatoid seaweed. Now, uh, while there was a property of antimicrobial agent in Kappaphycus alvarezi, especially, we also observed that the culture media used in Kappaphycus can hold growth-promoting ability. So we use the filtrates from key alvarezi culture and divide this into two sets. One is supplemented with low molecular weight organic compounds such as glucose, sodium uh, acetate, as well as a mixture of amino acids. And then there is also a group with no supplementation at all. So without supplementation, this is SSW minus. Uh, we observe growth promotion of the bacterial strain that were subjected to the filtrates in the different strains of bacteria. So here is W minus. Here also is W minus for another strains of bacteria. Another one here is W. So it shows that they have growth promoting ability, the filtrate from Kappaphycus culture. But then when supplemented with low molecular weight organic compounds, growth can be sustained for the duration of the observation period, so that all strains were promoted here compared to when there is no supplementation at all by these low molecular weight organic compounds. But nevertheless, it shows that our culture media, which contain exudates high in nutrient, could promote the growth of bacteria associated with the seaweed. Now, looking at the different conditions in two countries, like the Philippines, where I come from, and Zanzibar, where Dr. Misoya came from, we try to analyze the conditions in these two countries where the prevalence of ISIS disease is very high. So here in the Philippines, farmers tend to have, or there's a trend in shifting from the fixed off bottom method to floating line method, but then they still remain in the shallow waters. So shallow waters are still subjected to high water temperature, especially during low tide. So cultivars, even placed inside net bags, can still be afflicted with the ice disease and other fouling organisms. In Zanzibar, where the seaweed farms are located within the intertidal zone and the seaweed culture lines are touching the bottom, they are subjected to very high um, water temperature, uh, especially during low tide. And it shows here that the farmers can just wade in their farm without the swimming paraphernalia. So seaweed farms in Zanzibar are quite unique because the farmers there are mostly women, and they only need to walk within the shallow or during at high tide during maybe about waist deep water. Here, in this picture, it shows a remnant of the cultivar covered with some other seaweeds as uh, a competitor to the farm seaweed. And of course, um, hot summer months in Zanzibar, um, when these cultivars were examined closely, uh, both green and brown strains, they show very serious damage caused by ice disease um, in the two islands of Zanzibar, so namely Unguja and Pimba Islands. So these islands are collectively called Zanzibar. So the different places where we collected samples for ice analysis were found to be high in ice incidence of up to about 40%. And in Pimba, the ice incidence could be up to more than 70% in the different farms. So it shows that Pimba has relatively higher ice incidence compared to Unguja Island. This is during the hot, dry season from February to March 2017. And when the wet season arrives between May and June 2017, in both islands, the incidence of ice disease tend to have diminished by as much as 99%. Um, so in Zanzibar, this significant reduction in ice disease was an observation every time there is heavy rain in the islands of Unguja and Pemba. So we can say that uh, rainy season is a blessing to the seaweed farmers in the islands of Zanzibar. Now, in the case of epiphyte infestation, uh, epiphytes can disfigure the seaweed cultivars. In this case, this is Eucheuma denticulatum, which is loaded with the filamentous red epiphyte Polysiponia neosiponia complex, the upper photo here. But sometimes the cultivars can have another type of epiphytes. In this case here, there is also Ictocarpus, a filamentous brown alga. This is the appearance of the cultivars with Ictocarpus epiphytes. And then also um, cyanobacteria or the blue-green algae, Lingbia, which can be stranded or which can also entangled in um, the seaweed cultivars. This lower right photo here is a photo of a healthy Eucheuma denticulatum cultivar. So it's quite different when you have epiphyte infested cultivars compared to the healthy cultivar. So it could be observed within the same farm that you have mostly epiphyte infested cultivars and have also healthy cultivars. Now, taking a closer look at this infected thalli, heavy infestations of epiphytes can compete with light of the cultured species. This is the close-up of Polysiponia neosiponia complex here, showing the cystocarp. And you have also ectocarpus here, showing the sporangium. And here it shows you the another blue-green algae, oscillatoria, which macroscopically they can appear like dark outgrowths or tumor-like outgrowths in Eucheuma denticulatum. So this is a close-up of oscillatoria here. So epiphyte infestation can be very serious cause of damage to the seaweed farms in Zanzibar during hot, dry season. But then during wet season, the incidence of epiphyte infestation is much diminished, as shown here in this graph. So this is during the hot, dry season, where the number of epiphytes per centimeter of thallus can be as high as more than 30%, 30 epiphytes per centimeter of the seaweed in Unguja, but then relatively lower in Pimba. But still, epiphyte infestation is quite common in both islands of Unguja and Pimba. And during the wet season, incidence of epiphytes is much reduced um, uh, from May to June 2017 when we visited these farms in Zanzibar. So it clearly shows that both epiphytes and SIS disease can have very serious problem in the islands. Okay, just to show you the close-up of heavily infested uh, talus of Kappaphycus alvarezi with Neosiphonia epiphytes. This will not mean the end of the seaweed, but during rainy days, the epiphytes are gone and leaving behind only the poor-like appearance of the talus, but still continue to grow during the cultivation period of the farms in Zanzibar. A very interesting observation was made on a new strain of Eucheuma denticulatum that was introduced again to Zanzibar from the Philippines. So when these cultivars were cultivated over seagrass beds, it shows a very clean cultivars after a few weeks of introduction. Showing you here epiphyte-free cultivars over seagrass beds, in contrast to cultivars grown over bare sand, where the cultivars are heavily loaded with epiphytes. It seems that on bare sand, very high light intensity trigger the growth of epiphytes on the Eucheuma denticulatum strain. So this is quite interesting because we need to assess whether seagrass beds really can have positive uh, effect when cultivars or even the other seaweed species can be cultured here over seagrass beds. But of course, there are trade-offs to be considered when culturing over seagrass beds because of the role the seagrass beds also play within the ecosystem. Now, the immediate solutions after this observation is that doing away with the shallow water, the fixed off bottom culture method, and go to the deeper water methods of farming, like the floating long line and the rough method using a bamboo, um, can help mitigate the conditions of ice as well as epiphyte. That is, in this case, we have to move farms from shallow uh, waters towards the deeper waters. But of course, there's always a trade-off when farmed in the deeper waters. There's also this tendency to be exposed to the rough seas, like in the case of Zanzibar. So they place the cultivars in tubular nets, while in the Philippines they place it in small net bags. But that could answer the problem of fish grazing as well as biofouling with other marine organisms. We also make recommendations that seaweed farming should be done on a seasonal basis. So seasonal activity with implementation of a closed season during hot summer months, while the farmers can be engaged in other income generating activities like making seaweed-based products as value addition, like making seaweed soap, making seaweed shampoo or seaweed lotion, and even food products like seaweed salad. So this can be income generating to our farmers during lean season. Now, in conclusion, after this discussion, um, farming of eucheumatoids in farms more exposed to stressful factors is no longer producing viable crops that could sustain an industry. Global warming exacerbated the already stressful conditions of temperature, irradiation, and salinity that could drastically fluctuate over a short period of time during low tide, for example. Ice disease and epiphytes will continue to wreak havoc of seaweed farming in tropical waters, already beset with stressful and other anthropogenic factors like pollution and siltation. And farming in deeper waters is a viable option to minimize the impact of ice disease, which is a shift from the traditional fixed off bottom method. So with this presentation, I would like to thank again the organizers for inviting us in this conference. Thank you very much and have a nice day.